Greetings, pre-AP nerdlings. Today, we're going to talk about part 2 of microscopes and the cell. Stay tuned. Well, welcome back. Today, we're going to be discussing part 2 of microscopes in the cell for pre-AP. So if you're in pre-AP and you don't see pre-AP right here, you're on the wrong lecture. All right. So, introduction to cells. Cells are the basic units of organisms. Cells can be observed under the microscope, and there are basic types of cells. We have animal cells, plant cells, bacterial cells. We also have protist and fungi cells as well. So here's a picture of an animal cell with a lot of organelles that we'll be talking about later. Centrosomes, peroxisomes, microvilli, lysosomes, mitochondria, plasma membrane, Golgi apparatus, ribosomes, so on and so forth. So things that are found in plant cells but not in animal cells are chloroplasts, a central vacuole and tonoplast, a cell wall, and plasmodesmata. So here we have our plant cell. Like I was saying, plant cells have a cell wall that helps maintain its rigidity and maintain its shape, and they also have chloroplasts, as well as a central vacuole or tonoplast that animal cells do not have. And if you remember, chloroplasts are the sites of, if you said photosynthesis, you're correct. So limits to cell size. Why are cells so itty-bitty? The lower limit of cell sizes is bacteria, and the smallest bacteria are called mycoplasmas. Mycoplasmas are 0.1 to 1 micron long. Most bacteria are 1 to 10 microns. The upper limit, meaning the larger of the cells, are eukaryotic cells. Eukaryotic cells range from size to 10 to 100 microns. 1 micron equals a micrometer, and 1 micrometer is 1/1,000,000th of a meter. Just to give you a point of reference, a human hair has a diameter of approximately 20 microns. So what limits cell size? Surface area to volume ratio limits the size of cells. As a cell gets bigger, its volume increases faster than its surface area does. Smaller objects have a greater ratio of surface area to volume. So what cell organelle governs this? The plasma membrane. And why is a huge cell creature not possible? Well, it would take way too long for substances to get in. So imagine yourself on a flying reindeer. Yeah, that's right. Get ready for Christmas. We're riding a flying reindeer. Our flying reindeer, or Rudolph, is going to be flying at approximately 25 miles an hour. He's kind of a slow flyer. So if Rudolph wanted to get from Sam Rayburn High School to Houston going 20 miles per hour, it might take him about 2 hours. Now if Rudolph continues to go 20 miles per hour, but he wants to go to Austin, it's going to take him a lot longer. Probably something around the terms of, 10 to 15 hours. He is still flying at the same rate, but it's going to take him longer to get there. Just like it's going to take substances longer to get to the middle of a large cell and carry nutrients there than it is to take those nutrients to a small cell. So if you look here, cells with a high surface area to volume ratio are going to be easier for all of those solutes and nutrients to get into and out of the cell. The diffusion rate itself is still the same. Just like when we're flying on Rudolph, he's still going 20 miles an hour. It's just going to take him longer to get to Austin than it's going to take him to get to Houston. The larger the cell is, or the lower the surface area to volume ratio is, the longer it's going to take to get nutrients into and out of the middle of the cell. So one of the ways that cells have solved this problem is to colonize, meaning we have tons of tiny little cubes that form next to each other in the shape of a larger cell or a big blob, but they're still separate entities. This allows for those nutrients to pass into and out of the cell at faster rates. So the rate of diffusion of materials will remain the same as cell size increases in size, meaning it doesn't matter if we're riding Rudolph to Houston or if we're riding Rudolph to Austin. Same speed, 20 miles per hour that we have to go. So it means that in a large cell, it's going to take a lot longer for needed materials to reach the center of the cell and to exit the cell. So limits to cell size, metabolic requirements set upper limits. In a large cell, we cannot move material into and out of the cell fast enough to support life. So we have our small cell, and different nutrients are moving into and out of the cell at a much faster rate than it would to go into a big cell, get to the nucleus, and then exit the cell. So how to get bigger? We become multicellular. The cell divides. And that's what I was talking about in the previous slide when I was showing you all of those little cubes. Number of cells. Organisms can be unicellular, meaning one, composed of one cell, or they can be multicellular, composed of many cells. We are multicellular. There are two different types of cells. We have prokaryotic cells, which are unicellular, meaning one. They have a cell wall and a cell membrane present. They lack a nucleus, and they also lack membrane-bound organelles. However, they do have ribosomes, which help them synthesize or make proteins. Examples of prokaryotic cells are bacteria, and bacteria are living. The second type of cell are eukaryotic cells. Most eukaryotic cells are multicellular, although some are unicellular, such as amoebas. They have a nucleus, which is where all of the genetic information in the form of DNA is stored. And they also have membrane-bound organelles, such as the Golgi apparatus, lysosomes, peroxisomes, and the endoplasmic reticulum. Examples of eukaryotic cells are plant cells, animal cells, fungi cells, and protist cells. So cells, or at least eukaryotic cells, have what we call organelles. Kind of like the organs in our body, cells have little bitty organs called organelles. They're very, very small in size, and they can only be observed under a microscope. And each of those has specific functions, just like each of the organs in our body has a specific function. And they are found throughout the cytoplasm. Cyto means cell, and plasm means a plasma-like fluid. So if you think when you go to donate blood, sometimes they want to take your plasma out of your arm. So I always think of cytoplasm like the cell's blood. So the first organ we're going to talk about in part 2 is the cell membrane. It's also called the plasma membrane, and it's found in all cells, whether they be prokaryotic or eukaryotic. The function of the plasma membrane is to transport and protect cells. They have a property called selective permeability. Selective meaning it's choosy, and permeable meaning something can go through it. So it's choosy about what it lets into and out of it. It is selectively permeable, again meaning it's choosy about what goes into and out of the cell. It has two layers of phospholipids. Phospho means it contains phosphorus, and lipids means that it's made out of fats. So our phospholipids. We have a phospholipid molecule with one polar phosphate head. So this is polar, meaning that it has a charge, and it's going to attract water. It's hydrophilic, meaning it loves water. And then it has a nonpolar tail that is hydrophobic, meaning that it's scared of water or water feeling. So hydro means water, philic means loving. So the head is hydrophilic, while the tail is hydrophobic, meaning fear of. Just like if you have a fear of spiders, you have arachnophobia. And people that like small children are called pedophiles because they like small children or love small children. So phile means to love or like, and phobic means fear of or scared of. Most molecules have to go through protein gates to get into and out of the cell, kind of like we have to use a door to get into the classroom. So cell membrane proteins. We have channel passageways into and out of the cell. These are donut-shaped little passageways or channels, and they're embedded throughout the cell membrane. They allow different substances to pass into and out of the cell. We also have receptor proteins that are embedded in the membrane. They receive messages or receive other types of substrates or molecules that the cell needs. They will then transmit the information into and out of the cell. They're shaped kind of like a boulder. We also have marker proteins. They're basically like name tags of the cell, and they're different for each cell type. The shape of the protein is determined by the position of the amino acid, and they help cells identify namesakes after a transplant. So meaning, if a heart cell was transplanted for some reason into your brain would still recognize that as a heart cell. It wouldn't automatically become a nerve cell. So it's kind of like a little tag. So stay tuned for part 3 of microscopes in the cells, where we're going to discuss the endomembrane system. I'll see you guys next time.